people beaming in today. Hi, everyone. I'm Rick Page. And among the joys that I get to have as your Dean of the Larner College of Medicine is to preside over events like this. This is a true celebration. This is the annual Larner Research Laureate Lecture as part of the Dean's Excellence in Research celebration at our college. And this is the lecture part that goes with the award that was given last year, which is our highest research award. We have so much to be proud of in our college and the research enterprise we have. And as we reflect on these events, it's interesting that we are able to spread this out over 3 days. On Monday, we saw the research showcase for many of our current graduate students and postdocs, as well as members of the UVM Center for Biomedical Shared Resources presented. On Tuesday, we had the state of our research enterprise presented by Senior Associate Dean Interim Richard Galbraith, as well as our celebration and awards ceremony, and a very nice event where a number of us were able to interact afterwards in the Hale Gallery. As we celebrate, I do want to acknowledge and thank everybody who's participated in our research enterprise this year, last year, and going back, and recognizing times have been tough. The pandemic was not an easy time, and we're still working our way out of it. There are issues in hiring that are national and other issues here that we have to address. It's always hard to get a grant. Research is difficult, but also really fun. We can't take away the pay lines being difficult. We can't take away some of the challenges of research. We do want to make things as easy as possible for you to be successful in your research and celebrate, as we should, the accomplishments of our college in the last year. Today, we have a very special treat, and that is to hear from one of our own, Dr. Janssen-Heininger, and we've worked through the Americanization. It's Janssen, but here I was told and given permission to say Janssen-Heininger, who is one of our stars, and you're going to be hearing about in more detail as Richard Galbraith introduces her. I would make note that she is among the newest university distinguished professor at UVM, and she's been a member of the Department of Pathology and Laboratory Medicine for over 25 years. She's internationally recognized for the work that she does, and you're going to hear about today. Today, her lecture is entitled Redox Medicine for the Management of Chronic Disease. To formally introduce our awardee today, I'd like to invite Richard Galbraith to say a few words. I should note that Dr. Galbraith is, as I said, Interim Senior Associate Dean for Research at our college. He's been on the faculty since 1995. He's served in a number of roles. We listed them yesterday, including being Associate Dean for Research from 2012 to 2014, Interim Senior Associate Dean for Research during 2014, and from 2014 to 2020, he was UVM's Vice President for Research at the time, actually, when I was being recruited. We call him retired, and he is Professor Emeritus, but I'm tremendously grateful to Richard for serving in interim roles, first as co-interim director of the UVM Cancer Center, as we were working to recruit Dr. Randall Holcomb, and now currently as interim Senior Associate Dean for Research. So with that, I get to sit down and listen for the rest of this session, but please join me in welcoming Dr. Richard Galbraith.
Oh, thank you, Dean Page. So my job is to introduce Yvonne. Yesterday, I had to go through an extraordinary amount of data in a short time. Today is even more challenging. When you look at Yvonne's CV, you see she does nothing in half measures. First of all, she has 5 names: Yvonne Maria Wilhelmina Janssen-Heininger. And that gives you a clue to the numbers game we're going to have to get into later on. She received her BA and MS degrees in 1988 and a PhD in 1993 from the University of Limburg in Maastricht in the Netherlands, and then moved here to do a postdoc in pathology from 1993 to 1996 under a chair who's returned today. Welcome back. And then she did the classical academic thing, is to work her way up through every single degree, every single rank of the professorship until she achieved full professor in 2008. She also served as vice chair of the Department of Pathology for research from 2017 to 2021. She has been on numerous national and international awards that have been given to her, which we just don't have time to go into. It would take up the entire hour of the lecture. I can give you some highlights. She was a university scholar elected in 2012 here at the university. She obviously is the Dean's Research Laureate, elected last year. And as Dean Page mentioned, is a university distinguished professor, the highest academic degree award that we can get. And there can only be 10 of them at any given time. So they really are the pinnacle of achievement at the university. Starting in 2002, she has been a member of multiple NIH study sections and currently is the chair of the National Heart, Lung, and Blood Institute Program Project Review Committee. And now we get into the numbers game. There's no way of doing this without just going through the numbers. 7 patents. Associate editor of American Journal of Respiratory Cell and Molecular Biology on 6 journal editorial boards, and has served as a reviewer for 24 different journals. She has been a member or the chair of the organizing committees for 48 national and international conferences. She's been a member or a chair of 26 graduate thesis committees. She's taught 6 courses in pathology on 33 separate occasions between 2006 and 2022. She has 55 current or past trainees in her laboratory, and she's been to 91 occasions, she's been a national and international visiting professor or invited speaker. She has 164 peer-reviewed publications, 16 book chapters, and is currently funded with an R35 Distinguished Investigator Award from NHLBI and an R56 from the National Institute of Aging and an R01 from the NHLBI. She has 2 R01s pending and has completed 16 R01s already. That's a lot of numbers. So the last thing I'd say before I ask her to come up to the podium is that about 25 years ago, I had a crazy idea that something that I was working on in the rat brain might have something to do with the molecules that Yvonne was interested in. So I asked her if we could collaborate. And of course I was wrong. And so that was the last I saw of her for a while, which is a great shame, because if I'd chosen a better subject, I might have been riding on her coattails even today. So Yvonne, welcome. And we ask you to come up and give your talk, Redox Medicine for the Management of Chronic Diseases. Yvonne.
Thank you very much for those kind words, Dr. Galbraith, and thank you for the nice introduction, Dean Page. Really appreciate being up here, and I have to say that I feel quite humbled to be addressing you all today. And I also have to say that I kind of feel like those numbers make me feel really old and tired. No, I'm just, it's absolutely a delight to be up here to share my story with you. So I'd like to share with you a little bit how I got here, and then also tell you where I'd like to go next, because I'm definitely not done with being here yet. But so I'd like to start just by asking for a show of hands, because we're talking about redox today. And so I would like to know from you, have you ever bought over-the-counter antioxidant supplements that think that they're going to be good for your health? Raise your hand if you did. That's fewer than I thought.
You're lying.
You're So I like that. This is what we're going to be talking about today. So I hope by the end of the talk, you'd appreciate the concept of redox biology. I hope to convince you that glutathione is not boring. When I started my career, I thought it was the most boring molecule. I never thought I would ever get into glutathione. But so hopefully I'm convinced you that glutathione is not boring. You might also consider taking antioxidant supplements. And I hope also that we can collectively recognize the value of redox-based drug development and testing. So let's see now how I got here. So as you heard already, I'm originally from the Netherlands. I was born in a little town, Elslo, which is just 10 miles north of Maastricht, where I went to school. And but before I got to Maastricht University, there's a different story to tell too. And that is when I was in high school, I always liked chemistry. And interestingly enough, my chemistry teacher told me that he predicted in the future I would be struggling with chemistry. And so he told me to drop chemistry from the curriculum. Now, it turns out that I had a hard time with that problem, with that concept, because I really wanted to go to medical school. I wanted to be a medical doctor because I wanted to help patients. And so he said, well, not a problem if you drop chemistry from the curriculum, because you can apply to medical school and then you can do remedial chemistry and you can still get accepted. And so turns out not so fast, because Holland has very strict criteria for admissions numbers. And so they had a cutoff. And so if you were missing chemistry or anything like that from the curriculum, you were not allowed to even submit an application. And so there went my opportunity to go to medical school. And so it turns out that I was not the only woman or girl who was told that. All the other girls in class kind of had a similar story. So a bunch of my classmates went to nursing school, and I figured, well, maybe that's a good career for me too. But I was rejected because I didn't have the motivation to become a nurse. Unfortunately, or fortunately, depending how you look at it. And so. But luckily enough, Maastricht University at that time had opened up a brand new curriculum on biological health sciences. And this was a really interesting curriculum, brand new, because it taught you the importance of environmental factors like toxins, nutrition for human health. It was very strongly focused on biomedical research. And it embraced a method of active learning, small group learning, where we sit in groups and we had a tutor. And interestingly enough, that one of us always would have to take notes, had to summarize the conversation and make like meeting notes. And so that actually was incredibly helpful, because you really had to pay attention because you were actively learning. And to this date, I think that's where I learned how to summarize discussions. And I think that's why the NIH appointed me on their study section, because I was the person who really could summarize the discussions. So that's really helping me to this day too. But what's really important is that the curriculum in Maastricht focused on biomedical research, where you got to formulate hypotheses and test them and learn about the research methods. And so I was hooked on research. Then did my master's, where I set up my own clinical study, where I drew blood and did learn about sister chromatide exchanges and mutagenesis testing. So we published that. And so that resulted in a master's degree. And so after that, then I ended up going to do my PhD, because I was not done with research yet. And so I became a PhD student. You have to apply to be a PhD student in Holland. And so Dr. Paul Bourm took me into his lab. He was interested in silica-induced lung disease. And that's how I was going to start my career, except 3 months thereafter, the lab closed and I had nowhere to go. Now, luckily, Dr. Bourm had met Dr. Brooke Mossman from UVM. And she graciously accepted me into her lab for 3 months to kind of figure out the scenario. And so, I decided that it would be a good idea for me to do an internship for 3 months in her lab. So that 3 months ended up switching into 5 months and then 1 year and 2 years. And here we are today. So I never really left Vermont, but for 4 years, I went back and forth between Holland and Vermont to do my PhD. And this is my PhD thesis. It was a very successful endeavor in that I published 12 peer-reviewed manuscripts with Brooke, 8 book chapters and abstracts, and loved the research opportunities here in the United States. I was not ready to go back to Holland. And around this time, I also met my husband, Peter, who is from Vermont, and his family is in Vermont. And I was not ready to leave Vermont yet at that time, obviously. And so I figured, well, Brooke had agreed to take me in the lab for an additional year. I figured I better figure out how to get my own funding, right? And so I wrote a Parker B. Francis Fellowship application for pulmonary research. I was awarded that, and that's where it started. And so the next thing that happened is that Dr. Ted Boville, who's sitting here today, I'm so glad you're here, knocked on the door and said, Yvonne, we think that you are ready to become a professor. You know, we want to give you a faculty appointment in the department. And I remember saying, it's like, I'm just starting, you know, I'm not ready for any of this yet. I said, oh yeah, you're ready. We're good to go. And that's how I started, which is just really crazy to think about all of that. So then I decided, well, I'm going to focus my research on redox. And so I'd like everybody to understand, what is redox? What is this redox stuff anyway, right? So redox refers to chemical oxidation and reduction reactions. They can go back and forth, and they involve oxidants. So what is an oxidant? It is a reactive molecule. Doesn't have to be very reactive. Oxidants come in different flavors, but they want electrons from other molecules and become reduced after the reaction. Now, a reductant works on the other end, and that's a molecule that's going to donate electrons and becomes oxidized after the reaction. So these go back and forth. And an antioxidant then is a molecule that neutralizes the action of the oxidants. And so, like I said, oxidants come in different flavors. Hydrogen peroxide, you know about. Bleach, you know about, and they act very differently, obviously. You could wash out your mouth when you go to the dentist with hydrogen peroxide. You would never do that with bleach because the chemistry is just much more reactive. And so when I entered this field, and this is again going back to working with Brooke Mossman on asbestos fibers, which have intrinsic oxidative properties. So the dogma is that oxidants induce oxidative stress. They cause damage. They catalyze reactions that are out of control, resulting from too many oxidants, highly reactive oxidant species, causing oxidative stress. And you have then reactions that result in damage to your DNA, lipids, and proteins. And so that's really the dogma when this field started. And I'm just realizing this doesn't quite all up, because there were just hunches from other domains in research that told us or pointed us to oxidants carrying out completely different functions. And that new domain is redox biology. So oxidants carry out reactions that are controlled. They're important for normal function, and these reactions allow you to carry out complex tasks, and they're important for embryogenesis, exercise tolerance, and marathon running, and immune defenses. And so the chemistry between here and here is obviously very different. And so if oxidants can carry out biological functions, then it would also go that perhaps antioxidants are not always good for you. And turns out that's exactly the case. In some settings, antioxidants can actually promote growth of tumors. And there was a famous, vitamin E beta carotene trial in lung cancer that had to be stopped early because there were many more tumors that were developing following the taking of the antioxidant supplements. Antioxidant supplements also decrease the beneficial effects of exercise. So, when you train to run a marathon, antioxidant supplements can actually undo the beneficial effects of the exercise. And antioxidants can also decrease lifespan and use. So that means that we have to think much more carefully about these reactions, these chemical reactions, whether they're good or whether they're bad. That's definitely more to this than what meets the eye. And this is where the molecule glutathione comes into play. So glutathione is a molecule that consists of 3 different amino acids: cysteine, glycine, glutamic acid, a tripeptide. And this is one of the major redox buffers in the cell. It's very tightly regulated, so glutathione concentrations are always constant, and they can range anywhere from 5 millimolar to even higher. And we studied the lung, and in lung lining fluids, glutathione is really high, 500 micromolars in the lung lining fluid, suggesting that it's there for a very specific function. And glutathione regulates redox homeostasis, and it was originally believed that when you have an excess oxidant burden, the glutathione becomes oxidized. It forms through a covalent attachment between the 2 cysteines, and that's all there is to glutathione. And when you talk about oxidative stress and measurements of oxidative stress, sometimes you see this in clinical specimens. People measure an increased oxidation of the glutathione redox couple. And so this is how glutathione works. Turns out not so easy. There's much more to that, and I'll tell you that in a minute. But I have to first take you back many years back when oxygen began to increase in our environment. That is actually when cysteines in the proteome also showed up in organisms. And it actually is interesting because if you look at the organismal complexity as it increases, so does the content of cysteines in our proteome. So cysteines in our proteome have co-evolved with the use of oxygen from our environment. And cysteines are very carefully used in proteins. They occur in just about every class of proteins. These residues are highly conserved and are really important in regulating structure and function. And cysteines are relatively underutilized. And so, again, as we increase in complexity, so does cysteine utilization in the proteome. So from a cysteine perspective, we are equivalent to cows and to mice. So, so it turns out what we have learned over the past 10 to 15 years, that a signal from an oxidant is transduced into a biological response through the oxidation of cysteines and proteins in very carefully orchestrated reactions. So cysteines can be oxidized, but not all of them are oxidizable. The oxidation is regulated and reversible, and there's a lot of specificity to this. And the type of cysteine comes in different flavors, and it really changes how the protein looks, and it regulates structure and function. And so cysteines are really the chameleon of amino acids because it can take on so many different shapes and forms. But look, here's our glutathione molecule showing up. So the glutathione that was known when I started my career as a conventional redox buffer can actually act as a post-translational modifier of protein structure and function. And this reaction is referred to as F-glutathionylation, the conjugation of glutathione to the cysteine in the protein. And so I want to this is the last chemical reaction that I'm going to show you. Just bear with me here. But you start with the reduced cysteine, it can get oxidized, and in the second step, glutathione then becomes covalently attached to the protein. So you're changing the protein with 3 amino acids, you're changing the size, you're changing the charge, you're really changing the function of the protein. And this reaction can happen spontaneously. It can also be catalyzed by glutathione S transferase. As its name applies, it transfers glutathione. Now, these glutathione S transferases classically had been studied as being important in drug metabolism, toxin metabolism, but it turns out they have a very different function because these glutathione transferases, these GSTs, they bind glutathione to transfer the glutathione onto the protein. Now, when I started my career, people told me, Yvonne, this reaction is thermodynamically impossible. It is not physiologically relevant. You only see this happen when a cell blows up and dies. And so it's like, well, this didn't quite add up for me. So I stuck with that course. So we have GST pi catalyzing this reaction. So we thought, and then on the back end of this reaction, we have glutaredoxin, which takes the glutathione off the protein. So you have a forward reaction and a reverse reaction, moving glutathione onto and off of the protein. And so this very much is, as I'm sure that you're all aware, we have phosphorylation reactions. And so you have a kinase and a phosphatase. So this works exactly the same, but not tackling serines or threonines or tyrosines, but in this case, the cysteines are controlled in that way. But so is there any relevance to any of this biochemistry to disease? Oh, I can hear myself.
1, 2, 1, 2. This one's definitely better. Sorry about that. Yeah, apologize.
Oh, there's always. So maybe we can have a quiz. And this sounds differently. So what are glutathione S transferases? What do they do? Did anybody pay any attention? Kirk. I can almost, yeah. You have the glutathionylation, which is catalyzed by GST, and then glutaredoxin do the reverse reaction. Very good. You got a solid B minus.
Also told
Should not have done that, right? Okay, are we good to go again? This actually sounds better. Okay, so I'm sorry for those of you who did not hear me. So when I started this research, nothing was known about any of this in disease, and including lung diseases. And so we were interested in tackling this in the settings of idiopathic pulmonary fibrosis, which is a fibrotic disease that is highly pathological. A lot of people die from this in the US alone. But I want to say that although we're studying fibrosis in the lung, also want to emphasize that organ fibrosis is really dismal. It's contributing to 45% of all-cause mortality in the US. So once you have a scar forming in an organ, the organ tends to fail. And so this can happen to the lung, this can happen to the liver, to the kidney. So we think a lot of these processes that we're studying in the lung in the context of idiopathic pulmonary fibrosis may also be relevant to other organs. And so what is idiopathic pulmonary fibrosis? It is, again, fibrosis occurring in the lung. You have a massive destruction of air spaces and scar formation. It's a disease that's associated with aging, and it affects a lot of people in the US with a median survival from the time of diagnosis between 3 to 5 years, a mortality similar to breast cancer. And so this is just what this looks like. Again, you have no normal alveolar structure left. There's a lot of scarring that's forming and a disruption of alveoli. So this makes breathing impossible. So we were interested in determining then how glutathione redox homeostasis is changing in these lungs. And it's been demonstrated already that fibrosis is a disease that's associated with oxidative stress. And I mentioned earlier that oxidation of this glutathione redox couple is measured clinically. And actually in idiopathic pulmonary fibrosis, this had been demonstrated. And if you looked at the mouse models of disease, if you lacked certain antioxidant systems or NRF2, a transcription factor that's regulating many antioxidant defenses, mice that were deficient in these were much more sensitized to the development of pulmonary fibrosis by various models, including asbestos. And actually, Brooke did some of this work on redox perturbations. And so then you would think, well, okay, if I then give more glutathione in the form of N-acetylcysteine, this cysteine precursor to glutathione, you can protect mice from the development of fibrosis. But large-scale clinical trials using N-acetylcysteine and IPF failed. So does that mean that we throw the whole concept of glutathione out the window? Well, I said not so fast, because we hadn't addressed all aspects of glutathione biochemistry. And so we actually developed a method to then be able to look at glutathionylation in the tissue. And so we took tissues, deparaffinized them, made them accessible, and did chemical tricks. And in this case, we use glutaredoxin, which removes the glutathione from the protein, to then label newly generated cysteines to determine, does glutathionylation occur within the tissue? And the answer to this question is yes, because if you looked at the non-diseased normal tissue, there was very little chemistry. But in the IPF lung tissues, there's a lot of glutathionylation reactivity, and we can quantify that. We can also do independent biochemical assays to show that glutathionylation, again, this form of chemistry of glutathione that had not been addressed, is actually massively increased in lungs from patients with IPF. And the higher the glutathionylation, the lower the lung function in these patients. So what then is going on? I've told you, what you needed to know about the forward reaction and the reverse reaction. Let's look at the glutathione as transferase in these lung tissues. And GST-pi is of interest to us because that's the GST family member that's highly expressed in lung tissues. And you don't need to be a pathologist to evaluate or to value what's going on in these lungs. So in the healthy lungs, very little reactivity. In the IPF lungs, there's a lot of GST-pi reactivity in areas that show rebronchialization and massive remodeling. So I was looking here, but my esteemed colleague, Dr. Callie Butner, said, no, we need to look here because you actually see reactivity in the areas from which the disease is really expanding. And so that became interesting. And so can we do something about that? And luckily enough, there was a company in California, Telic, that was developing inhibitors of GST-pi for the treatment of lung cancers or cancers, because GSTs were overexpressed in a number of cancers and believed to contribute to chemotherapy resistance. And so this compound, TLK-199 or Talintra, was made. We couldn't get our hands on it because the patent was active. And so we had to generate it ourselves. And luckily, we have a colleague in the Department of Chemistry, Steve Flemmer, said, this is easy peasy. I can make this compound for you, no problem. So he made us the compound. And we then wanted to test, well, let's induce fibrosis in mice, and let's give the compound at a time where we already have the fibrosis present. And so we were pretty bold, and this is with Vikas and Afi in the lab, and we delivered TLK once every third day over a time frame of a couple of weeks. And what we saw in these studies, that if we started the treatment here, well, if you look at the time where the fibrosis was developed here, we started the treatment, we were able to stabilize the disease when we used transforming growth factor beta model of fibrosis. And with bleomycin, a chemotherapeutic that induces fibrosis too, if we started to treat the mice when bleomycin-induced fibrosis had occurred, we could actually dampen that fibrosis. But in these models, we were able to stop the disease in its tracks, which was pretty exciting. So let's look at, there's a whole bunch that I'm not telling you today, but let's look at the enzyme on the back end. You know now that glutaredoxin removes the glutathione from the protein. And so what happens with that in settings of fibrosis? And so here we looked at glutaredoxin mRNA in fibrotic or non-fibrotic individuals, glutaredoxin protein, fibrotic, non-fibrotic. And we didn't really see very many changes or large changes here. But when we looked at the enzymatic activity of glutaredoxin, something seriously is wrong in these diseased lungs. The glutaredoxin activity was substantially decreased. And so, now think about this. You have more GST, you have less glutaredoxin. So you really are allowing that environment of enhanced glutathinylation to really persist. And we also noticed something really strange in that the glutaredoxin now was glutathinylated itself. And so suggesting that the glutathione becomes conjugated to the glutaredoxin itself in association with its inactivation. And we actually modeled this by directly taking the recombinant glutaredoxin, putting it in the test tube, glutathinylating it, and it drops the activity. So this is a classical example where enzymes can get inhibited through negative feedback regulation by their own substrate. And Dr. Liz Corticelli, who is sitting in the audience, is looking very closely now at all the individual cysteines of glutaredoxin, because only this one is required for catalytic activity. And she's actually mutated each of them individually or in combination to figure out how the glutaredoxin enzyme gets inhibited through its own glutathionylation. Because if we ever want to develop it therapeutically, we want to obviously prevent its inhibition so that it doesn't undergo the same fate as the endogenous enzyme. But we didn't know that way back when, or in 2018 or in 2016, I should say. So we decided to take the recombinant glutaredoxin, see if we could label it, and we actually could take the endogenous enzyme and deliver it successfully into the lung. And then we asked questions, well, not giving the GST-pi inhibitor, but the glutaredoxin enzyme. What would happen if we directly administered the enzyme into the lung when you already had the fibrosis? And so we did a similar trick where we induced the fibrosis using the bleomycin model and then administered repeatedly glutaredoxin over a time frame of 2 weeks to then ask the question, what happens to fibrogenesis? And what we saw is that we could restore some of the enzymatic activity that was lost in these fibrotic mice. We could also, over time, decrease glutathionylation. And this was pretty exciting because we could also dampen the amount of hydroxyproline, an indicator of collagen content, as a marker of fibrosis in the lung, suggesting that the glutaredoxin was reversing the fibrosis. The histopathology does not look completely normal, clearly, but there's a lot more collagen when we treated the fibrotic lungs with glutaredoxin compared to the saline control group. So this then means that there is an underappreciated role for glutathione in the orchestration of lung fibrosis via reaction with protein cysteines. So we see increases in glutathionylation in lungs from patients with IPF in association with increases in GST-pi and decreases in activity of glutaredoxin. And I've also shown you that manipulating of either pathway allows us to control the extent of fibrogenesis. And so that means when you think about therapeutically, it's not about putting more glutathione in the already full bucket of glutathione, but rather what we need to think about is to control the reactivity of glutathione with protein cysteines. And so that's a very different way to think about redox-based treatments of chronic diseases. Now I want to shift focus just a little bit because in many conditions where you have fibrosis, these organs also tend to be much more prone to the development of cancers. And one unifying feature of cancer is actually an enhanced oxidative environment. And tumor cells have to adapt to that enhanced oxidative environment to allow them to survive and to allow them to metastasize. So shifting in redox focus is critically important for tumor biology and potentially also cancer management. And so does glutathionylation then, I've talked about glutathionylation and fibrosis, does it play a role in lung cancer as well? And here we don't have as many answers because these are areas that the lab is getting into more recently. But what's known is that lung tumors that have oncogenic KRAS, they increase their glutathione pools to survive. That's absolutely essential. Now, lung cancers are treated with platinum-based compounds, sometimes in combination with immune checkpoint inhibitors. But platinum-based compounds have high toxicity, limiting effectiveness, and tumors develop resistance to platinum treatment too. Guess what? Increases in glutathione and GSTs are associated with cisplatin resistance. And Brian kind of just brought to my attention the fact that cisplatin reacts with GSH, and the cisplatin glutathione adduct directly interferes with glutaredoxin activity. So that provides us with a really strong rationale for looking at this chemistry in lung cancer homeostasis as well. And so here we worked with a team of people. That's the beauty of being in the LCOM. There's lots of people who are willing to work together. So with Dr. David Seward, Matt Kinsey, and Joost van der Velde, we looked at the TCGA database and asked, what about these GSTs and glutaredoxin? Are they any different in lung cancer compared to adjacent normal tissue? Look at your GST pie that I've been talking about. This is sky high in lung adenocarcinoma compared to the adjacent tissues. GST is much higher expressed compared to any of the other GSTs. Whereas glutaredoxin in some of the tumors is substantially decreased compared to the adjacent normal tissue. So suggesting again that you shift that balance of glutathionylation chemistry towards enhanced glutathionylation. But is this in fact the case? And so here, Dr. Joost van der Velde took 106 tumors of patients and they made tissue arrays from the tumor as well as the adjacent normal tissue. And then asked the question, what happens with glutathionylation? And what happens with GST pi? And it's really interesting because half of the tumors that he looked at were high for protein glutathionylation. And it's really interesting because it's the same tumors that are high for GST pi. So if you express GST pi, you increase glutathionylation. So does this matter? And we think that this definitely matters. And here we again use that same TLK199 compound, and this had been used clinically with some therapeutic benefit in patients with myelodysplastic syndrome. But we induced tumors and using a KRAS G12D knock-in model. And so at the time that these mice became affected by the tumor, so a couple of weeks after we induced it, we began to administer TLK199 compound. And you can see right away that there's a big difference when you treat these tumors with TLK99 compared to the KRAS G12D model. So these tumors now are much smaller. The tumor nodules in number are the same, but the size is substantially smaller. And these mice live 1 month longer than the non-treated tumors. And in the lifespan of a mouse, 1 month is substantial and something to pay attention to. Now, what about glutaredoxin? So in this case, we did studies where we looked at wild-type mice and mice that lacked the glutaredoxin gene, and we induced then the tumor, the KRAS G12D oncogene, in the glutaredoxin deficient background. And so now you see right away that the tumor nodules are much greater. We had to sacrifice or euthanize these mice earlier because they got so sick. You look at protein glutathionylation in the absence of glutaredoxin, this is much higher compared to the wild type animals, and there's more tumor nodules. And this was also done by Dr. Reem Aboushusha, who is also sitting here in the audience. And what's also really important and relevant for working within the LCOM, and one of the advantages that we have is that we have great access to clinical specimens. And what Dr. Matt Kinsey was able to do through fine needle biopsy, when the tumor is being sampled for clinical diagnosis, he provided some extra tissue to Dr. Joost van der Velde, who then grew tumor organoids. And so now we can test, what's happening with the biochemistry of the tumor directly in these 3D cultures. And what we did with these tumor organoids, we directly administered glutaredoxin as a potential treatment. And what we saw is that we could increase the levels of glutaredoxin. And here we saw something really interesting. And so in that when we administered glutaredoxin, glutathione levels in the tumor decreased. And remember what I said, tumor cells, they have to elevate their glutathione pools to survive. Glutaredoxin is doing something really strange in that it is decreasing glutathione pools. And actually, Dr. Reem Aboushusha developed a brand new pathway that controls glutathione levels in lung cancer. So we're really excited to pursue that. But we then also, remember what I said, cisplatin is the chemotherapy that's used clinically as first-line therapy in lung adenocarcinoma. And we wanted to see, well, what happens if we co-treat these tumors with cisplatin plus glutaredoxin? And this is actually really cool because in these lung adenocarcinomas, this is the cisplatin-induced killing curve. And you can see that we can shift this to the left with glutaredoxin. We also see this in the mouse KRAS G12D organoids, where we administer a virus expressing glutaredoxin. We can also shift that curve to the left, suggesting that we might be able to treat tumors with cisplatin in conjunction with glutaredoxin to induce cisplatin-induced killing. And so we're really excited to pursue that now in a new NCI, hopefully funded R01 project. So, so what I've shown you with the bottom line is that the story that I told you with fibrosis is also highly relevant for lung cancer. And that glutathione-dependent protein cysteine oxidation may be highly relevant for lung cancer too, based on the observations that we have a lot of GST-pi that's upregulated in these tumors. Glutaredoxin expression seems to decrease, and I've shown you that that closely regulates glutathione homeostasis and cisplatin-induced killing. So we hope to be able to translate some of this forward. So again, that shows that you don't want to just change pools of glutathione, but really want to think very specifically about controlling the specific targets of protein oxidation by controlling this kind of cysteine-dependent protein biochemistry. So is this relevant for prime time? Can this be used clinically in treatment? And it actually turns out that cysteine-based drug development or redox, these cysteine redox-based drugs are blockbuster drugs. When you think about Tecfidera, this is non-specifically, but it reacts with cysteines, and this is a blockbuster drug in multiple sclerosis treatment. These EGFR and KRAS G12C drugs, they are engineered specifically against redox active cysteines, and they are FDA approved, and they're changing the course of disease. So pharma thinks we're ready for cysteine-based redox drug development, but are we ready in Vermont to embark on cysteine-based or redox-based drug development? And I want to point out that we are well known around the world for thinking exactly about those questions. And I want to highlight this because there are a number of individuals, some are in the room, who are thinking a great deal about that and have come up with really interesting patent or patent applications, potentially new compounds to target these kinds of pathways with a great potential for further therapeutic development and commercialization. And I want to give a shout out to Brian Cunniff because his work in the lab when he was a graduate student with Nick Hines now has resulted in a compound that's targeting mitochondrial redox homeostasis that's currently in phase 1 clinical trial in malignant mesothelioma. And I could not be more proud of those endeavors. And I'm so excited to see what's going to come from that. But I think in Vermont, we are ready to think about an institute for redox medicine. And I think that there is a number of components that we've already been touching upon and have been developing. Medicinal chemistry approaches. I've told you the story about TLK-199. There's much more that can be done. Computational chemistry. We're working with our computational chemist friends and colleagues to come up with better structure, function oriented and designed drugs. Preclinical testing platforms. We have some of the best models here in Vermont. You may not realize it, but the animal models that we have developed, the human organoids, et cetera, are just really fantastic. We have a great intellectual properties already and commercialization efforts. And I think that these endeavors can be embraced much better to go forward and to position ourselves around the world, even more so. In particular, thinking about partnerships with Wake Forest University and Sendai. I was supposed to be in Sendai, Japan. I chose to be here with you today. So, but I could not have done any of this on my own. I have many people to thank. And I have viewed Nat Pointer, Jos van der Velde, John Alcorn, as Karina Klaas as the early cornerstones of my lab. They took the risks on me to enter my lab when I felt like I was not ready to start my lab. Tad Boville for offering me the faculty position and made it all possible. Brooke Mossman is just a fantastic mentor. Nick Hines, a great colleague. Mark, you're here. Susan Wallace, I'm so appreciative of you because I like being able to walk into your offices and say, help, what do you think? Susan is the one who actually kept me focused on science when I was raising kids and thought I had to quit because I couldn't do it all. She said, Yvonne, big mistake, stick with your career. And I kept doing this. Deborah, who is our chair, thank you so much for supporting all of our research efforts. Means a lot to me. Charlie Irvin is somebody who always has his office open, and Charlie is the one who really allowed me to pivot from environmental toxicology to think about human biology and introduce me to a lot of key people. And a lot of us are thankful for Charlie because he is the one who reinvigorated the Vermont Lung Center, and we have absolutely fantastic people and many leaders here thanks to Charlie's efforts. And Dr. Neil Wouters was a fantastic collaborator who allowed me to keep the connection with Maastricht University. And together we have graduated, I think, more than 14. That's another relevant number. 14 joint PhD students between our institutions. So I hope that we find a path forward in that arena as well. These are the people who I'm grateful for because my success in redox biology, I owe to them. Albert Vandervliet, my close colleague, I always run ideas by him and he tells me I'm wrong many times. And usually he's right. Vika Sanathi and Nikki Reinert, they both trained in my lab and now have their independent, very successful careers. I'm so grateful for them. In particular, Nikki, I started work on nitric oxide and nitrosylation, and she said, but Yvonne, you got to think about that glutathione molecule because it's doing the same thing as your nitric oxide molecule is. And so she is the one who caused that pivot from nitrosylation to glutathionylation. She was the one who discovered glutaredoxin. I was not paying attention. So the moral of the story is always pay attention to your trainees because they have better ideas than sometimes I get the credit for. And Nikki is just one of them. And I'm grateful to Nikki. Colleagues around the world, we are so well connected in redox biology and medicine. And so I've had the opportunity to collaborate and interact, most recently in Barcelona with many of the leaders. Bruce Freeman is another mentor. This is at a meeting in Oxford with Louis Narrow, who won the Nobel Prize for nitric oxide. Despite what I just said, I did not stick within that domain. Sorry, Lou. The people in the lab, it's because of you I get to do what I get to do, and it's because of all of you I get to be up here and talk about all of the work. But it's really, you know, your diligence every day. I'm looking at Liz, I'm looking at Reem. Sometimes the research is really hard and we can't find all the answers. I'm so grateful to all of you to stick, despite the adversity that we face, right? Experiments failing is because of your diligence and perseverance that I get to be successful, and I hope I'll make yours successful as well. So thank you all very much for all of that. And then last and not least, the most important contributors are my family, my mom and dad. I miss them both so much. They've done everything for me and enabled me in ways that I cannot even begin to tell you. My sisters, Jacqueline and Suzanne, with their joie de vivre, I think you can see that. I always love to visit them in Holland. My husband, Peter, who kept me in Vermont. It's his fault. He kept me in Vermont, and he's always looking for the next mountaineering adventure, just like some other people. And then my kids, I didn't think I was able to have a career in science while raising kids, and I did it. My daughter Mira is a very proud graduate from UVM a year ago, and I cannot believe that actually she is the one who placed my temporary crown last week, which is so cool, as she hopefully will get into dental school. In keeping with the tradition of the Heininger families, there's many Heininger dentists in the state of Vermont. So she's going to keep that tradition going. And my son Skylar, who is a sophomore at UVM, both in computer science and biomedical engineering. And he is already contributing to the ALCOM research endeavors because he's working in some of your labs. And so I'm incredibly proud of him too. And then lastly, all of the other people, my collaborators who I don't get to talk about today, my grant funding, I've been incredibly privileged to be so well supported through my colleagues who I've mentioned, and also through the NIH. They've been really good to me. So thank you all very much for listening, and I'm gladly answer any questions if you have them. Thank you.
Thank you very much. That's an inspiring talk. That's great. So we do have some time for some questions. So that people in the audience put their hand up. We have a microphone, and there are also some people who may be calling in with questions who are online. So who would like to step up to the microphone first? While people are thinking, let me ask you this. What's the specificity of the cysteine that allows it to be glutathione? I mean, there's cysteine all over the place. So what is it that makes it these particular targets that get glutathionated?
That's a very technical question. It's a very good question. No, what I didn't have the chance to get into is that these reactions are, very compartmentalized. And so it is likely, and I didn't go through that, but you have another oxidative event first. And so you have like an oxygen producing enzyme that is operating very close to the site of the protein that is oxidized. And so then, and you also have your GST sitting there kind of as a scaffold holding the whole thing together. And so by virtue of having a GST interacting with the target protein, having the oxygen producing enzyme in close proximity, can you catalyze those reactions? Just like a kinase and a phosphatase, you have scaffolding complex there too that govern whether something is going to be phosphorylated or not. So it works very similarly.
Okay.
It's just like we're less familiar with the chemistry.
Okay.
Yeah.
There's a question in the back there.
Yeah, Yvonne, really great talk. I had a question about when you add glutaredoxin therapeutically, you showed that the glutathione pool decreases. It seems somewhat counterintuitive. Is that because the glutaredoxin gets stuck in the glutathionated state? Or do you know why does the glutathione pool decrease in that case?
Yeah, that's again not something that we were expecting. It's actually the great news is that it's the topic of a new NCI grant, which is going to start next year. But that's actually the pathway that was discovered by Reem Abu-Shusham. And so this is linked to system XC-, which is a cystine glutamate antiporter that's highly relevant in cancer and prevention against ferroptosis. And so it turns out that glutaredoxin is controlling the activity of system XC-. And so it's acting on a very specific target that is glutathionated and then allowing cystine to be taken up. And so we can get into the nitty gritty, but it's an ovarian tumor deubiquitinase, highly relevant to system XC- regulation and a number of other targets that are relevant to cancer, immunotherapy, et cetera. But there's critical cysteines in that ovarian tumor deubiquitinase, which are being reduced by glutaredoxin to allow that uptake and the activity of system XC- to occur. That's a lot of information.
Okay, well, if there are no more questions, I know that Vice President Dombrowski wanted me to say he wasn't leaving because he only got a B-. He has a 1 o'clock meeting. So thank you very much. It's a great talk. Well done.